Hey everybody, we are back and I'm gonna bring up Gabriel Corso.
Hey,
Thank you so much for joining. We have had like a marathon morning and early afternoon of open source AI products, and so much of it has been around developer tools and what I think of as software. And I know that you are also building software, but I think it's a really different. I think it will expand how people are thinking about where open source AI is going. So I'm really excited to chat. Thank you for joining.
thank you very much, Matt. No, this is so cool. I was following the live earlier. So
Awesome.
many projects.
Well, let's jump into it. Maybe can you give your brief background and kind of maybe talk about your research that you're working on, and then I'll kind of I'll ask you questions. But I'd love for people to get sort of this one requires a little bit more of grounding, I think, in biology.
Sure. Yeah. No, I actually come from a computer science background, but after doing some research in undergrad, I decided, this was right after the AlphaFold 2 was published, and so I was really inspired by kind of how these advances in machine learning could really change the way that biologists would work. And so that's why I decided to do a PhD in this space of structural biology, which is the space of trying to study how kind of proteins, molecules kind of interact with one another and forms different shapes. These are critical problems for drug discovery and many of critical applications also in climate and health. And so, right after kind of AlphaFold 2 was published and I started my PhD, it was clear that, the next big frontier was modeling biomolecular interactions. So not just a single protein like AlphaFold 2 did, but actually understanding how different proteins, and different molecules interact with one another. And this is because, proteins in our body don't work on their own. If they typically interact with other molecules, if we want to design a drug, this is a small molecule that will go and, for example, bind a particular drug target and inhibit it. So, to develop a new drug, understand biology, we need to understand these biomolecular interactions. And so this has been kind of the topic that I've also kind of done my PhD in. We were the first ones to introduce kind of generative models for this task. We were the first to actually develop AI models that would outperform the traditional physics-based baselines, and that have been used in the field for decades. And most recently we released Boltz 1, which was the first open source model to achieve the level that was state of the art that was set earlier in the year by AlphaFold 3 from DeepMind, that however was kept commercially closed source.
So can you talk so you start out as a computer scientist, software developer. You get really interested in this topic and you kind of dig in. What is the hello world of AI in applied to biology? Is it like, can you just help us think about it as a more of a somebody more grounded in software and like language models and image models? I can picture how that works. Weather forecasting models, I can start to picture how that works with sequence prediction. What does that look like under the hood?
Yeah, I mean, the way that you can think about it is that these biomolecular structures that we are predicting, that AlphaFold or Boltz 1 are predicting, are really these miniature machines in 3D that to us, you can look at them, visualize them in a computer, and they look beautiful. But when you actually look at them, they are actually Armstrong of size. These are things that require to shoot X-rays to find out the structure from. So you're really looking at something that is 1/1,000,000th of a meter that we are able now to predict the structure. So it's really sort of like this thing that obviously relies on some incredible advances that biological engineers have done over the past century on how we are able to actually look into this deep into cells, and then once they get into the computer, they somewhat become like any other machine learning problem. Obviously, we have particular biases and particular things that we want to integrate into the models, but at the end of the day, I say that I don't really understand biology. I just play with molecules.
Interesting. Okay, so that kind of makes sense. As soon as you can take it and put it into, as long as you can put it into a file, it's not any different than a 3D model, or so? And then when you were, so can you talk us through what the process was of building the model? In my mind's eye, I always picture kind of, are you thinking about what the environment is? Like, in a weather model, you have kind of, let's take the degrees, and then let's take the radar, the blips, and let's take the pressure, and we're kind of doing next token prediction. But what did that look like for you when you were building the model?
Yeah, I mean, one of the biggest challenges in building these models is parsing the data. So this is, decades of, biologists, releasing their data that they generated as part of their studies. And, this data was, generated starting from the 70s for human use. So, when you actually need to use them, with a computer, you need to really parse this data. And, so one big challenge is, how do you parse this data and the models? Then, like any models, these models are particular kind of diffusion models. So, we've heard some examples of images and videos, that kind of models, from a very high level kind of generative modeling perspective. But, they have a lot of specific components and specific type of architectures that people have found to be very effective on this task. And then one of the other biggest challenges was obviously scaling. These are models that are not yet at the level of large language models that are used in natural language processing, but are much bigger than models that we are typically dealing with in an academic setting. And so this, to get a rough order of magnitude, this is models that train for around a month on GPUs for AlphaFold 3. So a lot larger. And so obviously when you work at that level of scale, first of all, for us, it was a big challenge at the start, accessing the compute, but then, obviously dealing with the scale that implies.
Yeah. So getting at the compute, did you kind of model out how long you thought it would take and how much compute you would need to start and how much it would cost? Or how did you is like that what the, grant proposals are basically?
Yeah, no, we did. And we actually started working on it even before, we had the compute that we knew we would need at the end. So, we started, because we didn't have the compute at the start, we started at a kind of smaller scale, building all the things that we required, but, testing it at a smaller scale. Then we initially kind of got this grant from the Department of Energy, where we were able to start a large. The issue with these grants and these clusters from the Department of Energy is that it's used by a lot of people. So, you maybe run for 2 days and then you queue for a week. And so, we could see, very, very strong initial performance. And so at that point, we actually kind of did the partnerships with a large startup, which provided us with the kind of the rest of the compute that we needed. And, in exchange, obviously they really wanted to access this model as soon as possible. So gave it to them.
Interesting. So you were able to do some of this at a small scale. Did your confidence level change as you were training? So you're cleaning the data. You have a rough sense of maybe what models you're going to use. Although I want to kind of, you talked about diffusers, but I'm sort of always curious. I know some people who've started with transformers and then realized that transformers weren't the right tool, or had to create some kind of judge to alter it. What were sort of the, I guess, step function increases in your confidence that this would work in a meaningfully better way than, you obviously, AlphaFold was out there, so you knew that it was possible. Was that a big piece of it?
Yeah, so largely, obviously, we add, from our research as well as the research of the rest of the field, a lot of understanding about what kind of architecture are more likely to work and what are less. And as you're saying, on small scale, you can test a lot of things, but you never really know kind of how you're gonna what kind of scaling you're gonna get and improvements you're gonna get when you're gonna scale 10x, 100x. And so that, we are really going a bit blind into the large run, to be fully honest. But obviously, once, this run started, the convergence was quite clear. And so we could really see kind of where the model was going.
So then you started to see where the model was going. You started to, I would imagine, do the evals, and you're like, okay, this is actually getting pretty strong. And ultimately, I think it was the Boltz 1 is that at parity with AlphaFold?
Yeah, so in many domains it is. There are like many different kind of type of evaluations that you can do. There are specific modalities where one model is better, but at the general level, it's a very kind of similar level of performance. So when we released it, people in the field were very excited and have adopted the model across the industry.
That's awesome. And then sort of separately from building it, you had to make a choice at some point I would imagine, of whether this is going to be open source. Curious how I saw it. It was. Do you release it open source and open weight, or open source only, or what did you do, and how did you kind of come to that decision?
Yeah, so we actually ended up going for open source, and that means inference code, training code, all the data pipeline that I was mentioning before to curate the data, as well as open data and open weights. So we really kind of released everything, and there was two kind of like decisions. The first one of why we would release the inference code and the model open source was there are a lot of biologists, whether in an academic settings or in large pharmaceutical companies and biotechs, they really they work on understanding and finding cure to diseases, and really want to access these models. So we felt it was really important to kind of give them something that they could use straight away. The reason for releasing kind of the training code and the data and so on was to really allow the rest of the machine learning community, people like us last year, to build on top of it, extend it. And this has really been incredible to see a lot of the contribution that the community has done to the model. Just last week there was a new design model that was kind of published open source, that was built on top of Boltz. NVIDIA has announced that they are kind of building kernels specifically for Boltz to really optimize its performance during training and inference. So it's really been incredible to see kind of the adoption and the support that we got by releasing things open source.
That's interesting, because it used to be, I keep transposing sort of open source today from open source from like 10 years ago. And 10 years ago, it would have been everyone's contributing to my code, and so my code is getting better. And it feels like today it's more ecosystem than it is actual lines of code. It's I'm going to fine tune on top of it, or I'm going to develop a kernel that runs this because it's useful. It's almost like the different layers of the stack participate, as opposed to everyone participating at the code layer. Does that seem true?
Yeah, I think it makes sense. And I think, largely the challenge here is that, these models are very large, can get trained, maybe once every 6 months or so. And so, obviously we have, now overgrown also the core contributor kind of size, where, the ones, building the core models themselves. But then, what the community is really interested in is, building, for, fine tuning for their specific applications, or, optimizing. And so we've seen a lot of, also companies, releasing, building something and then actually releasing it in our repo to everybody, because they don't necessarily see it as, their competitive advantage. And so, we're really kind of seeing, the start of this ecosystem, which is really incredible.
What's just sort of, I know we're about to be at time. Where do you see this going in terms of, is this, you have a unique product where like the actual manifestation of the research is it drug development? Is it understanding how different diseases work? Sort of what do you see as like the kind of end state as people adopt?
Yeah.
Other models.
I mean, the way that I think about it, what Boltz does is one kind of relatively small step of, the very complex pipeline that is, for example, drug development. And this pipeline is largely still done in, the physical lab. But I think, as I look forward the next 5 years, this pipeline will largely move in silico. And so with Boltz, we really want to expand, the impact that we have and the kind of different parts of the pipelines that we cover to really have a, end-to-end pipeline that biologists really want to try to create the new virtual lab for biologists to really make them a lot more efficient and more effective at, finding drugs, understanding biologies and so on.
Really cool. Well, thank you so much for joining today and for sharing a bit about Boltz. It's been an amazing project to watch, and we're excited to see where things go.